So very good evening all. Sorry for the interruption. First of all, I would like to introduce myself. I'm Arun Kumar, undergoing PhD in the Department of Crop Physiology from Tamil Nadu Agriculture University, Coimbatore, India. And I'm working under the guidance of Dr. M.K. Kalarani ma'am. And today I'm here to share few things about my research work, that is exploiting the potential of new plant growth regulator melatonin in the alleviation of drought and high temperature stresses in rice, that is Oryza sativa. So in this slide, I would like to express the reason for choosing this research work and what are all the research gaps here behind. First of all, abiotic stresses are the major concern for food and nutritional security. Among that, drought and high temperature stresses are predicted to increase to a higher extent, especially in tropical countries like our India. In recent days, plant growth regulators have been used to mitigate these abiotic stresses. One among that plant growth regulator is melatonin. So far, only few research works have been done in melatonin with respect to abiotic stresses. So the current study was framed to study the impact of melatonin on physiological, biochemical, and molecular aspects of rice, which is subjected to both drought and high temperature stresses. So far, we have seen research gaps. Now I can say what is melatonin. So what is actually melatonin? It is an indolic compound which is synthesized from an amino acid called tryptophan. This melatonin initially discovered in mammals. Later only it was discovered in plants. Here I am showing that melatonin can alleviate the negative impact of both biotic and abiotic stresses. Why? Because means it acts as a good antioxidant and reduce the reactive oxygen species. It also scavenge reactive nitrogen species also. Meanwhile, it also enhances the plant's antioxidant system. So it also acts as an antioxidant and it also activates the antioxidant systems. This is how it nullify the different biotic and abiotic stresses. Apart from this melatonin also activates the genes which are responsible for plants' stress tolerance. Hence, we may call this melatonin as a master regulator in plant defense. So coming to the application point of view, in mammals, it regulates circadian cycle, that is day and night cycle or sleep-awake cycle, and helps plant to cure disease like Alzheimer. In plants, its primary function is to serve as the first line of defense against internal and environmental, especially oxidative stressors. Then coming to the biosynthetic pathway of melatonin in plants, as I mentioned already, it was synthesized from a tryptophan, which is an amino acid. This tryptophan can convert it into tryptamine with the help of enzyme TDC. Then this tryptamine will convert it into serotonin. From serotonin only, we will get melatonin through two biochemical reactions from with the help of enzyme called SNAT and ASMT. So far, we have seen what is melatonin, how it is synthesized in plants, and its application value. Now we can see how this melatonin enhances stress-tolerant capacity of plants. Melatonin usually enhances stress tolerance by modulating the antioxidant system and maintaining relative water content through osmoregulation. And it also alters the expression level of stress tolerance genes and melatonin biosynthetic genes like SNAT and ASMT. Here we can see if the plant is subjected to abiotic stress, when we apply melatonin exogenously, it will enhance the endogenous melatonin level. This endogenous melatonin level may act as an antioxidant inside the plant system. So it may scavenge reactive oxygen species and reactive nitrogen species. Apart from this, it will also interplay with the plant's hormone system. For example, during stress time, ABA will be synthesized more in plants. If there is more ABA in leaves, means the stomatal will get closed. So only less photosynthesis happen. What actually this melatonin do means, it will down-regulate the expression level of ABA biosynthetic genes so that we may get less ABA content in plants during stress time. So we may enhance the water use efficiency of the plant with the help of melatonin during abiotic stresses, especially during drought stress condition. Then coming to the activities, in my research work, the efficiency of melatonin in imparting tolerance against drought and high temperature stress was assessed. We have measured numerous traits like leaf gas exchange parameter, which is nothing but photosynthetic rate, transpiration rate, and stomatal conductance. Then we have studied the interaction of melatonin with other hormones, especially ABA and GA. Then we have to study the expression level of melatonin biosynthetic genes like SNAT and ASMT, as I mentioned already. Then we have to assess how melatonin enhance the yield and yield components of rice during this drought and high temperature stress condition. So from this work, we can optimize the concentration of melatonin, which could mitigate the effect of both drought and high temperature stress in rice, and identify the physiological biochemical mechanism behind the stress tolerant capacity of plant with respect to our melatonin treatment. So far, we have optimized 200 micromolar as a suitable concentration for mitigating drought and high temperature stress in rice. Thank you.